Hello. So in this video we are going to talk about the detoxifying mechanism of our body. So let's start. Detoxification. It is the physiological or medicinal removal of toxic substances from a living organism, including but not limited to the human body, which is mainly carried out by the liver. So, detoxification is the physiological medicinal removal of toxic substances, removal of toxics which generally occur in liver in humans. So, in medicine, detoxification can be achieved by the decontamination of poison ingestion and the use of antidotes, as well as techniques such as dialysis and chelation therapy. We'll talk about it all of this in detail in the next slides. So, types of detoxification. What are the types of detoxification? These are the alcohol detoxification, the drug detoxification, the metabolic detoxification, and the alternative medicine. So, alcohol detoxification is, of course, when we remove alcohol from the body, right? The alcohol, those alcoholic people which ingest large amount of alcohol must be detoxified. That by this process, drug detoxification, the druggies, the drug addicts need to be removed the drugs from their blood, from the body by this process. The metabolic detoxification occurs in all of us in liver, where the harmful waste materials within our body need to be removed by this process, metabolic detoxification. An alternative medicine is something like a therapy, which we'll talk about. Now, let's start with alcohol detoxification. So, alcohol detoxification is a process by which a heavy drinker's system is brought back to normal after being used to having alcohol in the body continuously. Serious alcohol addiction results in a downregulation of GABA neurotransmitter receptors. Alcohol detox is not treatment for alcoholism. It is not the treatment. After detoxification, on the other treatment must be undergone to deal with the underlying addiction that caused the alcohol use. Right, so that is about alcohol detoxification. Now, drug detoxification. Drug detoxification. Drug detoxification will reduce or relieve withdrawal symptoms while helping the addicted individual adjust to living without drug use. Drug detoxification is not meant to treat addiction, but rather it is an early step in long-term treatment. It's just the drug detoxification is just a medicinal process by which we remove drug from the body or the blood. It could not cure addiction of drugs. So detoxification is achievable drug-free or may use medications during treatment. Drug detoxification varies depending on the location of treatment, but more detox centers provide the treatment to avoid the symptoms of physical withdrawal to alcohol and other drugs. Most also incorporate counseling and therapy during detox to help with the consequence of withdrawal. Metabolic detoxification. Now this is the main topic we need to talk about, the physiological changes that occurs in our body for this metabolic detoxification. So an animal's metabolism can produce harmful substances which can then make less toxic through reduction, oxidation, conjugation and excretion of molecules from cells or tissues. This is called xenobiotic metabolism. Now our body during metabolism could make many harmful substances like peroxide ion. It could be made within our body and it is a toxic substance, so it needs to be removed. So how that happens? By metabolic detoxification. By the reduction, oxidation and conjugation, these harmful substances could then make less toxic materials, like peroxide will then be converted into something less toxic. So this is metabolic detoxification or xenobiotic metabolism, where xenobiotic materials, the harmful material is converted into less harmful materials. Now, because it is the primary mechanism for ridding the body of xenobiotics, foreign chemicals, the metabolic detoxification is getting rid of the harmful materials, xenobiotics, which are the foreign chemicals. Enzymes important in detoxification metabolism include cytochrome P450 oxidases, UDP-glucouronyltransferases, and glutathione S transferases. So these are the three very important enzymes, which is cytochrome P450 oxidases, UDP-glucouronyltransferases, and glutathione S transferases. These are the three enzymes which are very important in metabolic detoxification. Let's see how and what function they does. So the liver is the primary detoxification organ. It filters blood coming directly from the intestine and prepare toxin for excretion from the body. Significant amounts of detoxification also occurs in the intestine, kidney, lung and brain, with phase 1, 2 and 3 reactions occurring throughout the rest of the body to a lesser degree. Now what is this phase 1, 2 and 3 of detoxification? We are going to study in the next slide. So the detoxification has three phases: one phase, second phase and third phase. Three phases of metabolic detoxification. So in this slide we could see very clearly. So there are the toxin which are fat soluble. Toxins are fat soluble. So it undergoes phase 1 detoxification, where oxidation, reduction, hydrolysis, hydration, dehalogenation kind of reactions occur upon these toxins. And what it forms is an intermediate metabolite which is slightly water soluble. Right, these are fat soluble toxins and they undergo these reaction under phase 1, and then they become intermediate metabolites which are slightly water soluble. Then they undergo phase 2, where these undergo glucouronidation, sulfation, glutathione conjugation, acetylation, etc. Reactions, and forms excretory derivatives which are completely water soluble, and these products are finally excreted out. They are excreted out by phase 3 detoxification, right? For example, the water soluble non-toxic substances or wastes can now go to bile, where they will go to intestine and then will be excreted with feces or stool, or they could go into the blood serum, from where they would go to kidney and be filtered out as urine. So this is the whole detoxification process. So let's see this again. So there are the toxins, or what we call the xenobiotics. Toxins undergo phase 1 detoxification. Phase 1 detoxification. These were fat soluble. Now they become intermediates which are slightly water soluble, and then they undergo phase 2, where they become the excretory products, and by phase 3 of detoxification they are excreted out, right? So this is the simple so toxins were fat soluble and they are intermediates. After phase 1 the intermediates are slightly water soluble, and after phase 2 the excretory products are completely water soluble, H2O soluble, and then by phase 3 they are completely excreted out. So let's go to the next slide. So let's start with the details of the three phases of detoxification. So phase 1, detoxification. This process is the enzymatic transformation. So what happens in phase 1 detoxification is that under most circumstances, phase 1 enzymes begin the detoxification process by chemically transforming lipid soluble compounds into water soluble compounds. Like I said earlier, in phase 1 the lipid solubles or fat solubles are converted into water solubles right, And preparation for phase 2 detoxification. So this is the phase 1 detoxification. The bulk of phase 1 transformation reactions are performed by a family of enzymes called the cytochrome P450. So the first phase, one phase of detoxification is mostly controlled by P450 cytochrome, P450 enzyme. Let's see what it does. So cytochrome P450, let's talk about that. Cytochrome P enzyme are relatively non-specific. Each has the potential to recognize and modify countless different toxins. Cytochrome P metabolizes toxins very slowly compared to other enzymes. For instance, compare the predominant cytochrome P34A, which metabolizes 1 to 20 molecule per second, to superoxide dimutase, which metabolizes over a million molecules per second. So like it says, the cytochrome P is very slow, right? So cytochrome P's are very slow. For example, the comparison is given here that it metabolizes 1 to 20 molecules per second, while another enzyme, superoxide dimutase, metabolizes over a million molecules per second. So it is very slow. But for major sites of detoxification overcome the slower speed by producing large amounts of cytochrome, which may present up to 5% of total liver proteins, and similar large concentration can be found in intestine. So because this cytochrome is slow, what they do, they produce a large amount of cytochrome in the liver, large amount or a high concentration of cytochrome is produced to overcome this slowness or this tardiness of the cytochrome, right? Now examples of cytochrome P's, some of the cytochrome P's which are found in our body are the flavin monooxygenase, which is responsible for detoxification of nicotine from cigarette smoke. So we know that the cigarettes have nicotine in them, which is a harmful toxic, which are detoxified by flavin monooxygenase enzyme, and this is a cytochrome P enzyme. Now alcohol and aldehyde dehydrogenase is which metabolizes drinking alcohol, right? So this is another enzyme, another cytochrome P enzyme. Now monoamine oxidase, which break down serotonin, dopamine and norepinephrine in neurons, are the largest of several antidepressant drugs. So these antidepressant drugs are also cytochrome P's. Now let's go to phase 2 detoxification of enzymatic conjugation. So phase 2 detoxification is enzymatic conjugation. Conjugation means if this is A, this is B, they are joined together, so this is conjugation of them, right? So this reactive intermediate is still unsuitable for immediate elimination. So after phase 1, the intermediates that had been formed, are still could not be eliminated like that, right? This reactive intermediates that had been formed after phase 1 is still unsuitable for immediate elimination from the cell for a couple of reasons. Activation of phase 2 enzymes is responsible for the anti-mutagenic and anti-carcinogenic properties of the metabolic detoxification system. So in phase 2 what happens? Anti-mutagenic or anti-carcinogenic, anti-cancer toxins are removed in phase 2. Let's see how that happens. So again let's go back to this and let's see. We have studied about phase 1 already, what are the reaction happens, and this intermediate formed, it is still not ready to be excreted. It could not be directly excreted at this stage, right? It is still harmful for our body. So they undergo the phase 2 reactions where conjugation reaction happens, and then they convert them into excretory derivatives which could be excreted out. So let's see how that happens. NFR2 At genetic level, the production of most phase 2 enzymes is controlled by a protein called nuclear factor erythroid derived 2, NRF2. Nuclear factor erythroid derived 2. So the phase 2 enzymes are controlled by this NFR2 gene. However, the presence of oxidative stress activates NRF2, allowing it to travel to the cell nucleus. Then there is oxidative stress within the cell due to free radicals or something. So that activates NRF2. That tells this oxidative stress is generally due to free radicals. Free radical, for example, this is a free radicals. Oxidative stress occurs, right? And these free radicals are the toxins, right? It tells the cell that it has toxins within it, and so NRF2 is activated. So after NFR2 is activated, what happens? NFR2 regulates the activity of genes involved in the synthesis and activation of important detoxification molecules. Certain dietary constituents including sulforaphane from broccoli and xanthohumol from hops may also directly activate NRF2 and stimulate antioxidant enzyme activity. So for antioxidants, we require antioxidants to remove the free radicals from our body and reduce the stress within our cell. And for that, if we eat broccoli, which contains sulforaphane, it will activate NRF2, and thus the antioxidant will be produced and the radicals will be removed. Now some of the important enzymes of phase 2. Important enzymes of second phase detoxification. First is UDP-glucouronyltransferase. So UDP-glucouronyltransferase catalyzes glucouronidation reactions, the attachment of glucouronic acid to toxins to render them less reactive and more water soluble. So what the UDP-glucouronyltransferases does is that it if this is a toxic material, it attaches glucouronil with this toxic material, binds glucouronil with this toxic material, so it becomes less toxic, less reactive and more water soluble. So that is the function of glucouronyltransferases. Another important enzyme of phase 2 detoxification is glutathione S transferases. It catalyzes the transfer of glutathione to phase 1 products. Like I said, again what it does is that if this is a toxic material, this enzyme, what it will do is it will attach a glutathione to this toxic material so that it becomes less toxic. Glutathione S transferase play a major role in the metabolism of several endobiotics including steroids, thyroid hormone, fat-soluble vitamins, bilirubin and prostaglandins. Sulfonyltransferases. What sulfonyltransferases does? They attach sulfates from sulfur donor to end or xenobiotic acceptor molecules. If this is a toxic material or xenobiotic acceptor, these enzymes attaches a sulfate, SO4 2 minus, right, sulfate. They will attach a sulfate to this toxic material to make it less toxic and more water soluble, because phosphates are water soluble. Another important enzyme is arylamine N-acetyltransferases. N-acetyltransferases detoxify carcinogenic, which is cancer causing, and aromatic amines and heterocyclic amines. Right, so that is its function. And now we go to phase 3. So phase 3 detoxification is transport. In phase 3, no reaction takes place. Instead, only transport of the excretory materials takes place, and they're excreted out. How that happens? Phase 3 transporters are present in many tissues, including the liver, intestine, kidneys, and brain, where they can provide a barrier against xenobiotic entry or a mechanism for actively moving in xenobiotics and endobiotic in and out of cell. So their main, like I said, the main function is transport. They are present in many tissues like the brain, the intestine, the kidneys, right? So they are present in the brain, the intestine, the liver, the kidneys, right? And what they do is provide a barrier against xenobiotic entry or removing the xenobiotics out of the cell. Now, phase 3 transporters belong to a family of proteins called the ABC transporters, which means ATP binding cassettes. They need ATP, right? So they need this energy in the form of ATP because they require chemical energy in the form of ATP to remove the toxins or they remove the excretory materials, right? They need ATP. So these are the phase 3 transport, phase 3 detoxification. So again we come back to this picture again and see, after phase 2 excretory derivatives have been produced which are completely water soluble, and in phase 3 what is happening is that they are removing these water soluble excretory derivatives either through bile, which goes to intestine and then removed with the feces or stool, or they are going to serum, which will be filtered into the kidneys and removed as the urine. So this is phase 3, where there are different transporter which require ATP transporters which remove these excretory derivatives out of the body. Thank you. We need to study about the alternative medicines as well. So alternative medicine. So this is the last type of what is called detoxification. Certain approaches in alternative medicine claim to remove toxins from the body through herbal, electrical, or electromagnetic treatment, such as the aqua detox treatment. I think this is a load of bullshit, but anyway. So there is no evidence for toxic accumulation in these cases. The liver and kidney automatically detoxify and excrete many toxic materials, including metabolic waste. In alternative medicine, you know what they do is kind of it has no evidence actually that it actually works. What they do in alternative medicine is they use snake stones, body cleansing, and Scientology purification, and Gerson's therapy, oil pulling, food patch detoxification, nada. Anyway, so thank you for watching and please like and subscribe the channel.